Welcome to the next lecture on the methods in microbial ecology with reference to environmental biotechnology. And in this particular lecture, we are going to talk about the culture independent analysis methods, particularly the fingerprinting based methods, which enable us to perform the community profiling in any kind of environmental systems through two important approaches, which are the ARDRA and DGG. So, the ARDRA or Amplified Ribosomal DNA Restriction Analysis, the first one that we are going to discuss in this class is basically this will allow us to identify the phylotypes present within a community through preparation of the clone library and then subsequent restriction enzyme digestion of the 16S rRNA gene fragments which are actually cloned from the environmental sample. So, as you can see here, the name implies this is the amplified ribosomal DNA. So, the ribosomal rRNA gene is amplified from the sample and then it is cloned in order to segregate the different species member or the phylotypes into different clones, and then each of these clones are actually used to obtain the gene insert within their plasmid, and they are subjected to restriction endonuclease digestion, and then followed by restriction endonuclease digestion, the profile that we get that is actually compared. So, the intrinsic assumption in this case is that environmental sample consists of numerous microbes. So, each of the few micrometer of the samples will have several thousands organisms or several hundred organisms. So, when we take a sample, we take multiple subsamples of these samples, extract the DNA, total DNA, and that represents the total DNA. That means the DNA should be representative of all the organisms present here. And once we have this total DNA with us, the DNA is subjected to the PCR application, or that is the PCR of 16S ribosomal RNA gene, using the universal primer or taxonomic group specific primer. Now, what is universal primer? Universal primer is a primer pair which is optimized to amplify all bacteria. So, irrespective of its taxonomic identity, all bacteria DNA 16S ribosomal RNA gene will be amplified or potentially be amplified by this set of primers. While there are some group specific primers. Group means taxonomic group specific primers. Suppose I want to only amplify Pseudomonas, or I want to amplify only the gram positive bacillus type of bacteria. So, similarly, there are many taxonomic group specific members which could be used similarly, and also there is rather a primer set for archaea as well. So, there could be bacteria specific primer and archaea specific universal primer, or there could be a broad primer, universal primer, which will amplify all kind of prokaryotic 16S ribosomal RNA gene. So, once we have that PCR reaction, we are going to have a kind of, if we run the sample on a gel, that is the agarose gel, we will get a band, and it is a clear band because 16S based on the primers that we have selected, a single size will be amplified, and of course, this might consist of the 16S RNA genes from all the different members. Suppose in this case, if you have 8 or 10 different members, so all the members 16S RNA genes are amplified. So, ideally within this single band, all the amplified products of this are included. Now, here we go for the cloning process, and this cloning of this rRNA gene will be done using a standard microbiological or molecular biology cloning procedure using a plasmid vector. So, each of the plasmid molecule, the linear molecule of the plasmid will be ligated with this PCR product, and the concept is one plasmid will be able to take only one PCR product of the 16S RNA gene, and it will circularize. So, ideally one circular plasmid circularized upon the insert is present. Insert means the 16S ribosomal RNA gene in this case is inserted properly. So, it is circularized, and when it is successfully transferred through the DNA transformation experiment. So, as you can see, the blue white colony selection is used here. So now the positive clones are picked up, and all these positive clones, suppose I am able to pick up 100 positive clones out of these the color selection based method. So, all my 100 clones are supposed to have each of these 100 clones will have individually the plasmid inside them, and each of these plasmids are having one 16S RNA gene insert. So, if we believe that each of these 100 clones are having a different 16S, just we believe that, or we take it for example, then there should be actually 100 different phylotypes or 100 different species. But in reality that may not be the case. We may find out that a particular phylotype or the species was more abundant. So, if we have taken 100 clones in a clone library, we may find that actually 20 clones are containing their plasmids having the same 16S RNA gene insert, and rest 80 clones are having some different types of 16S RNA gene insert. It depends upon the relative abundance of the 16S RNA gene, and also it is also connected to the original relative abundance, of course, and it is also partly connected to the lysis efficiency, because all the cells may not be lysed with equal efficiency. So, some cells might lyse very well in the lysis condition, and some cells might be reluctant and they need some harsh condition maybe to get them ruptured and the DNA to come out. So, it requires some kind of optimization. So, the scientists who work on these kind of things, they know that the type of samples they are using. So they optimize the protocol so that the maximum cell lysis and a well representative clone library can be constructed. So, it is ideal that as many as possible clones, positive clones are selected, so that we can have a broad coverage of the different types of ribosomal RNA genes. Now, once we have the clones with us, these clones, suppose in this case I am showing you a data with more than 100 clones. So, each of the 100 clones are having one plasmid. So, the plasmid DNA can be extracted, and then can be another round of PCR can be made to re-amplify the 16S from the plasmid, and then that PCR product will be subjected to restriction enzyme digestion. So, as we digest the sample with restriction enzyme, based on the sequence composition of the 16S ribosomal RNA gene insert derived from the plasmid, if they have similar type of sequence, they will show a similar restriction digestion pattern. But if they don't, then possibly they will cut at different sites and essentially will lead to a different type of restriction patterns. So, like you can look at this any of these digestion patterns, and you will be able to find out that somewhere you have quite a similar type of, like these two particular clones. If you see here, they are having almost similar type of digestion pattern. That means these two 16S RNA genes are likely to be similar. Similarly, if we see there are some other positions are there, like this one, this is also similar to the other one. And you can see this in this set of clones, we have some other clones which are having a digestion pattern of very distinct digestion, like these two clones are similar, while these two clones are quite similar with respect to their digestion pattern. So, we look for this similar digestion pattern and we group them. So, as we group them, we possibly reach to the phylotype type of information, that we group the similar digestion pattern containing clones and their data together, and if needed we can have actually more number of digestion, so that it's not a single enzyme digestion. Separately, ideally we should digest with 3 enzymes, and then we take the data that which clones are having a similar pattern, restriction digestion pattern, even after digestion with 3 separate enzymes in a separate digestion experiment. Because if 2 inserts are same in with respect to their sequences, they will produce similar patterns with different enzymes even. So, the number of phylotypes, that is the unique restriction enzyme digestion pattern in this case, are identified, and the clones representing the different 16S ribosomal RNA gene phylotypes are sequenced, and the 16S gene sequence obtained then are analyzed for their phylogeny and taxonomic affiliation, and then the community composition is obtained. So, I will give you here one example of our own study, where we worked with just 58 clones we could pick up in that sample. We are just optimizing and standardizing all these things and seeing how it is doing. So, we could have this data where 58 clones are there, and you can see one particular type of restriction pattern is found to be most abundant. So, 8 out of 58 is having that particular pattern. 8 clones or 8 plasmid are having the same pattern, while some other patterns are moderate and rest, and it followed the pattern like this. Now, some representative clones are sequenced for the 16S ribosomal RNA gene. Now, as we sequenced or keep on sequencing these representative clones from this, we are able to identify through a similarity search in the database, NCBI database, that the first most abundant one belongs to the firmicutes. That's a group of bacteria. The second is the acidobacteria, third is the nitrospirae, fourth is again firmicutes, next one is bacteroidetes, and so on and so forth. So, we can have actually up to genus level information if our sequencing quality is good, and we intend to do so. So, this is one method that is very regularly followed, and I will come later in this lecture with some case studies that how this is being actually used. So, the second one is a very interesting method where we do not apply the cloning based procedure in the beginning. So, rather we try to segregate the different phylotypes or different species based on their DNA composition or DNA based sequence. So, this is called denaturing gradient gel electrophoresis, separating very similar genes. So, 16S ribosomal RNA gene, for example, is a very similar gene. So, it is like in a pool of 16S RNA genes which are amplified. You can assume that there are many 16S RNA genes are there, but they are coming from different bacteria. So, it is expected that they might be different in their sequence because they are coming from different bacteria. So, different bacteria would necessarily have a different sequence. Now, how do we segregate them? Now, one fundamental assumption again, that each of these different 16S ribosomal RNA genes actually coming from different bacterial species or taxa, they have different base sequence composition. They are all same gene, but they have some variations in the nucleotide base composition. So, now if we expose them, these DNA fragments, that is 16S RNA genes, to a range of different conditions which actually allow the DNA to be melted, like to denature. So, each of the DNA will show different melting behavior. Melting means denaturation behavior. Like the DNA which will contain higher number of hydrogen bonds, like with because of the presence of more GC, possibly will require higher temperature or higher denaturant concentration, rather than the DNA which is having a low GC percentage or low GC concentration. So, this is one of the methods to resolve the phylotypes, is this denaturing gradient gel electrophoresis, which separates the genes of the same size, because all are of same size, because we have used the same primer for the amplification, that differ in their melting profile because of the difference in their base composition. Now, by DGG, DNA fragments of the same length but of different sequences can be separated, and generally the length of the 16S RNA gene that is amplified through PCR is 200 to 500 base pair, and the forward primer in the PCR reaction contains an additional 35 to 40 base pair GC reach sequence at the 5' end, in order to segregate and to have a sharp bands on the gel later when we run it on the gel. Like for in this case, as you can see that this is the first round up. So, we have maybe organism wise we have some separate organisms, if you can see, and one is the mixture of these organisms. As you can see that based on all our 16S ribosomal RNA gene, the same region is amplified, but because their 16S RNA base sequences are different, some is forming band over here, while some other is forming band here, and if we mix together, we will be able to have a banding pattern like this. So, this is a kind of an ideal DGG banding pattern. Now, the separation in this case of the phylotypes or different 16S is based on the melting behavior, and melting behavior depends on the base pair composition. However, the GC clamp ensures that at least part of the DNA remains double-stranded. I will explain this because if we allow them to denature completely, then the formation will be like the two single strands will be formed, and in the gel we won't be able to detect them. So they will migrate very fast then. So we do not want to lose them, rather we want to have a distinct band. So GC clamp is used so that they denature, but a small part of them is not denatured because of the very high GC content, and then they stay, rather the structure is formed like that they won't move in the gel anymore. Like to elaborate this, like this is the DNA double helix structure, and as you know, some GC bonds are more stronger. So if you have more number of GC in your sequence, it would require higher concentration of the denaturant or the temperature. So suppose we have a DNA, double-stranded DNA, and we expose it to higher temperature. Slowly we increase the temperature. We will see that the double-stranded structure will first start melting, and this melting will initiate at the AT-rich region rather than the GC-rich region, because the GC-rich region, these regions will contain three hydrogen bonds rather than the AT-rich regions, and eventually to break the GC bonds, you need to raise the temperature to a significantly higher temperature, and then they will be altogether to separate out to single strand. So that is called the melting behavior. Now in this case, we try to actually use this information that if we melt up to this single stranded, then possibly you won't be able to detect them. Rather, if we have some structure like this, using the primers, not at the actual DNA part of the DNA. So we use the primers which has the very high GC content. So that part will not melt, but the rest of the part will melt. So it will form a kind of a Y-shaped structure. So this Y-shaped structure, because of this structure, it won't be able to move in the gel and it will stay there. And later when we stain the gel, we will be able to see this. So basically the GC-rich region of the primer is used as a clamp. Okay. And the result of the DGG analysis of PCR products obtained from a microbial community is a band pattern, and the number of bands is a rough estimates for the microbial diversity of a given system. So ideally, if we have like 4 different types of 16S ribosomal RNA genes having 4 different types of GC content, then upon the DGG analysis we can have actually 4 different bands. So I will come to the picture, then you will be able to see it. So here we are actually using some different environmental DNA. So these are all the total community DNA from different samples, and as you can see that the DGG patterns are varying significantly. And now if I say that these samples are actually from a very extremely low pH to moderate to high pH, then possibly that makes some more sense, that with respect to changing pH or changing other environmental conditions, how the banding patterns are shifted. So you can see that some bands are consistent, while in sample numbers are varying. Some bands are consistent, while some bands are appearing in some other samples. So this might indicate the presence of some groups which are present only in some samples, or some groups which are consistently present in more number of samples. So ideally, each of these bands can be obtained, or the bands are cut with a knife or a sharp blade, and they are processed, excised, and these excised bands are used to further amplify, clone and sequence, so that we know that what is there exactly in this band. So ideally, this band could be made up of 16S ribosomal RNA gene fragment of multiple organisms. Why multiple organisms? Because it is because of the same GC content they have stopped there. That does not mean that it will contain only one bacteria. So one band is not indicative of one bacteria or one species. Rather, it could be multiple species whose 16S RNA genes in this case are having same GC content. So based on that, we need to actually further, in order to, if we want to analyze more, we can actually excise this and then clone and sequence it. So what can we know from the DGG? So we can actually know the community complexity, because more number of bands will reflect the complexity. Identify the community members by sequencing. Individually, each bands can be cloned and sequenced. Distribution of microbial populations inhabiting different environments like temperature, pH, etc. Particularly in water distribution system or different type of other effluent processing system. These kind of methods are found to be very useful because it's the same water flowing through different treatment processes. So how the treatment process is actually working on it, or a similar type of environment, but there are certain differences with respect to some parameters like the monitoring the community changes. So it has a very broad application, and the advantage is it is reliable, reproducible, rapid, and somewhat inexpensive in nature. Multiple samples can be analyzed concurrently. So the current practice is that we can actually run around 15 to 16 samples at a time, or even more than that. It is particularly useful to detect population change, addressing the question of stability and dynamics of microbial community. For environmental biotechnology purpose, this stability and monitoring the community becomes an important question to answer. So DGG was very successfully used before the advent of the next generation sequencing method, which was found to be more precise, more deep, able to identify or characterize almost all the organisms present there. So DGG remains a very popular method in order to characterize the community composition and community dynamics. And if this is actually done with ribosome, with RNA instead of a DNA, then it will actually reflect what type of organisms are active in this case. Now here I present some of our studies that done using the DGG and clone library method. So we can see that how the clone library method can give us the community composition, and I can take you to another. So we can have the three different type of mine tailings. We can see that how the community is varying from the different type of mine tailings which are present in a uranium mine. And similarly, there are other numerous papers where we see that the PCR, DGG and the clone library both are very popularly used in different wastewater effluent and different other processes which are having like low grade copper sulfide ore, bioheap, leachates, or maybe different other environments like succession of the communities within a very particular environment. So for this part, for the general text or the methods that we have discussed, the Brock's biology of microorganism will be sufficient. Otherwise, the papers that those are being shown may be consulted for some more details. So in conclusion, the concepts of two major community fingerprinting methods like the amplified ribosomal DNA restriction analysis and denaturing gradient gel electrophoresis DGG, those are discussed and their applications are highlighted. Thank you very much.